Okay, guys, good morning. Welcome to the class. Let me turn this on. All right, you guys made it before the midterm. Congratulations. That's the first step to success. Okay, so what we're going to do today is we're going to go through microanatomy of eukaryotic DNA, basically talking about the human genome most of the time. Okay? All right. So I'm going to take over the class for a little bit. So we're going to talk about genomes and gene expression in the next couple of weeks. Mark Siegel is going to take over to go over the molecular genetic analysis. Then I'm going to be with you for the rest of the semester most of the time. Okay. So before I start, I actually want to start with a question that I get asked every year, year after year. And it is this question: how can I do better at MCB? It usually comes right after midterm 1, so pay attention. So the answer to this is, let's review how we learn, okay? So our brains are wired to forget. If I ask you what you had for breakfast for today, you'll probably remember what you had, right? How about yesterday? You will remember. 3 days ago, maybe. 13 days ago, definitely not, right? So what our brains do is it purges all the unnecessary information, right? And the necessary information also goes out with it. All right, so if you basically plot, since we're scientists, we like to plot things, right? If you plot learning as a function of time, when you learn things, you start remembering things very easily, right? And then with time, you forget. What happens when you're working on MCB, in which it's going to be a mixture of memorizing terminology, learning concepts, and building up the molecular biology knowledge, right? And what we have throughout the semester is we have, it's a semester-long learning process. Let's say that we're plotting this semester in the x-axis again and the learning on the y-axis. And let's say for the semester you did nothing and then you studied in a rush, right? Which is very typical. So what you would do is you're learning a little bit, right? And then something is happening and you're studying and studying and studying. Then you're not doing anything, right? And then you're working a little bit and then studying a lot. So what are these question marks for? Exams, right? So this is the typical curve we see a lot with people studying for MCB. And this doesn't work because you're going to forget everything you learned right away because you don't actively recall what you learned. What's active recall? Active recall is kind of like how you would memorize a poem, right? You don't memorize a poem by reading it constantly, right? The way that you would memorize a poem is you would read a few times and then you would try to recall what you read, right? So that's how you actually learn, is you actively recall what you actually heard. So what are those active recalls? Those recalls need to happen throughout the semester, and these happen through some of the actions that you're going to take. And these actions are already listed in your syllabus, which you should do, which is to read the assigned material. It's very important because in the lectures we do cover a lot of material, but we do not cover it in some depth that you might actually need, right? We teach to the average class. We don't teach to individual people. So you need to read your textbook. You need to come to the lectures. Don't use the recordings as a proxy for lectures. Coming to the lectures is the best way to learn, and I'm so happy that you guys actually did that. You need to think about your study questions, and you need to go to your recitations and talk to your recitation instructors. And when you do that, what will happen is that your learning curve is going to stay up. You're going to start remembering things much, much better. And this is really important because by the time you come to the final, everything is cumulative. That means if you keep up the good work during the semester, you're not going to have to cram for the final, which never really works. Okay? So on that amazingly enlightening note, we're going to go to the lecture. All right. So in this lecture, what I want to do is I want you to get this idea of DNA as an information. We've spent a lot of time in POB and then in earlier the semester learning about the chemicals and the molecules that make up the cell, the molecular machines that copy the cell, etc. Right? But at the same time, we can think of DNA as an information. The DNA sequence has the information to encode for the proteins and all of the cell components. Okay? So what do I mean by that? What I mean by that is if you do an experiment in which you know the DNA sequence of a person, right? You take that DNA sequence, you put inside a human cell that doesn't have it, that you have removed its own DNA. That DNA sequence should program that human cell to develop into a human, right? Because it should have all the information from a person that would make up a person. So this is not a very good experiment to do, right? There are lots of ethical reasons why you may not want to do this. But you can do an experiment. What you can do is you can actually take a bacterial cell, right? Get its DNA sequence, and then in vitro, that means outside the cell, synthesize the DNA with its building blocks, right? These are nucleotides. Put it back into another cell that you removed its DNA from and see if now that DNA sequence that you synthesized, synthetic genome that you made, can convert that cell into its own identity, right? And this is an experiment that actually was done. So in this experiment, what the authors did, they took a, let me see here, Mycoplasma myocidis genome, and what they did is first determine its DNA sequence. So they isolated the genomic DNA, they sequenced it, and they put it into a database, right? There are databases that contains the genomes of many, many, many species. But what's amazing is what you can do is you can take that sequence, you can synthesize the genome in vitro from its building blocks, you can put it together through a carrier, in this case a yeast, and then you can transplant it into a new cell that used to be Mycoplasma capricolum, and now turn it into a Mycoplasma myocidis. So this basically is a kind of a proof of principle experiment showing you that the DNA has the information to make up the cell. Okay? All right. So then it shouldn't shock you then the genome size. The genome is all of the DNA inside a cell, right? The genome size roughly correlates with organism complexity. So very small bacteria like Mycoplasma have very small genomes, and as the organism gets more and more complex, such as the mammals, the genome size increases because it contains more information, right? All right. So the information is organized roughly, in very general terms, into genes. So a gene is defined as the entire nucleic acid sequence that is necessary for the synthesis of a functional gene product. The gene product could be a protein. It could be a non-coding RNA that works as a functional product. Okay. So. Again, it's not that shocking to you that the number of genes also roughly correlate with the complexity of an organism, right? But not perfectly. What you can see here is that, for example, a small organism like the flu virus has very few genes, and bacteria has a little bit more, etc., and humans have a lot more. And this water flea here has the highest known number of genes in the on Earth so far. It's about 36,000. All right, so it roughly correlates with the complexity of the organism roughly correlates with the number of genes, but also it correlates how you use them. So think of these genes making products, and think of the products as Lego pieces, right? And how you put them together actually makes the difference between different species, roughly, in general terms. Okay, and in this, in the rest of the semester, we're really going to learn how the information in the DNA is used. So we're going to spend a lot of time about how the DNA is used by the transcription machinery to make the mRNAs, how the mRNAs are used to make proteins, and how each step is regulated. And that's what I mean by how you use them. Each step is regulated so you can put the Lego pieces in different forms. All right. So we're going to talk about human genome now. And eukaryotic genomes, like the human genome, are organized as linear chromosomes. So our genome is broken up into linear pieces that are called the chromosomes. The human genome is about 3.3 billion bases. It has 22 autosomes. Autosomes are chromosomes that are not the sex chromosomes. Any chromosome other than the sex chromosome is called an autosome. So we have 22 autosomes and 2 sex chromosomes, X and Y. The females have 2 X chromosomes and the males have an X and Y chromosome. So we have a total, each of us have a total of 46 pairs of 46 chromosomes. That's 23 pairs. All right. What's in this genome, right? What's the information? What kind of genes are encoded in the genome? So here's a table that kind of summarizes this. We're going to go through each of these classes and learn what they are. Okay? So just to start with, here we have protein coding genes, which make up about 25% of our genome. So a transcription unit in this case is exons plus introns, okay? But not all exon is actually protein coding. Can anyone tell me an exon that's actually not protein coding? You learned about gene structure already, right? What is it? UTRs. Perfect. It's untranslated regions, right? These untranslated regions of the mRNAs are not coding for DNA. The exon is actually the RNA that ends up in the mature mRNA. That's the definition of an exon. A coding sequence is the sequence that encodes for a functional product, like a mature tRNA, transfer RNAs are a functional product, or a protein, right? So if it's coding for a protein, we call it coding because it codes for the protein sequence. Excellent. Okay. Let's see here. So we're going to talk about the protein coding genes. These can be, the protein coding genes can be from a simple transcription unit. So it could have a region of the genome that codes one product, okay? So in this case, this is kind of like a typical yeast gene, for example, which you should know by now, right? So what we have is for each gene, we have a transcription start site. So the transcription starts at this point, and the RNA polymerase tube transcribes the gene until it hits a terminator where it's going to stop transcription. And after the transcription start site is going to be the translation start site, which is typically an ATG codon. And the ribosome is going to translate this mRNA until it hits the stop, right? And here is, for example, 5' UTR. And this gene is going to contain promoter sequences that are going to be bound by transcription factors that are going to be important to recruit RNA polymerase II for transcription. And it's going to have some enhancer elements, which are DNA sequences that are bound by transcription factors, again, that regulate the RNA polymerase II binding to the promoter of the gene and the transcription. So this is a simple transcription unit. In humans and a lot of complex eukaryotes, we have complex transcription units. That means that the unit is actually split into exon and introns, and over 90% of our human genes are complex transcription units. So in this case, what you have simply is that the gene, the coding region of the gene is split into a certain number of exons. And sometimes these exons can be spliced together in different combinations, which we call alternative splicing, right? And due to alternative splicing, one complex transcription unit can give result to multiple products. So far, this should be more of a review for you. For example, in this gene, all of the exons are put together to create an mRNA that has this information. And for example, if the exon 3 is skipped in alternative splicing, then you would have a protein that has this information. And then if the exon 2 is missed out, you will have a protein with this information. So you can produce different products from alternative splicing. Okay. All right, so the protein coding genes, we classify them into usually two classes. One is called a solitary gene. These are genes that are found in only one copy. An example of this is the chicken lysozyme enzyme. This lysozyme enzyme is also found in humans. It's in our saliva. And what it does is it cleaves polysaccharides in a bacterial wall. It's also in our tears. So when we cry, we have some lysozyme coming out. So these are the examples of solitary genes. There are also another class of genes called the duplicated genes. So these genes are basically members of gene families. So these gene families can have several hundred copies. An example of this could be immunoglobulins. You have hundreds of copies of immunoglobulins in our cells, or olfactory receptors. These are receptors that are in our noses that can sense all kinds of different smells. All kinds of different, not smells. What do you call it in English? What? Smells good? Yeah. Right. Thank you for that. All right. So how do we have these gene families? Okay, how do they arise? They arise due to gene duplication. Okay, so here's an example of a gene family, and it is the tubulin gene family. So what happened in millions and millions of years ago, there was only one copy of this gene in an ancestral cell. And what happened is that this gene kind of got duplicated. So there was a second copy arose in the genome, and each copy started specializing for a different function. Okay, so they started diverging from each other. Each of their sequence started evolving differently. And so what we ended up with is after millions of years, we have now two copies of this gene. One is called alpha tubulin, and the other one is beta tubulin, with slightly different sequences and slightly different functions. Okay, so when this ancestral cell started evolving into two different species, these two different species are now independent of each other. So the sequences of this alpha and beta tubulin in these two species are going to evolve differently. What that means is they are going to accumulate mutations and differences differently from each other. But at the end, when we look at these two different species, we're going to see 4 copies of the tubulin gene, right? Each species is going to have 2 copies. Each species is going to have an alpha tubulin and a beta tubulin that are going to look like each other. And then these 2 genes are also going to look like their counterparts in species 2, but some slightly different. All right. So what do we call these genes in these gene families? When we're talking about a gene family within a species, such as alpha and beta tubulin, we call these genes paralogs. They're paralogous to each other. They're in the same species. They're similar, but they arose due to duplication or multiplication. And if a gene is compared between 2 species, but the gene is essentially the same, such as alpha tubulin in humans and alpha tubulin in mouse, we call these orthologous genes. They're called orthologs. Okay, so these are the same genes, but in 2 different species. And all of these genes, orthologs and paralogs, are called homolog to each other because homolog means similar. They're originating from the same original gene in the ancestral cell. Okay, so those are mostly protein coding genes. And in the genome, where they can be found is they can be found close to each other, but their sequences are slightly different due to how they diverge during time. Okay? So now there's another class of genes in the genome that make up a very small percentage of the genome, but it took a pretty large part of this genome table because they encode for very important genes. Okay? These are tandemly repeated genes, and the perfect example of this is genes that are encoding for ribosomal RNA. Without ribosomal RNA, nothing survives because you cannot translate, right? So although they make up a small portion of the genome, they're taking a large portion of our energy explaining them. All right, so how are they organized in the genome? They're tandemly repeated. What tandemly repeated means is you have the same gene sequence one after another, okay? So, but these are exact same sequence. They're not like alpha tubulin and beta tubulin, right? So there are many, many copies of it. They're tandemly located. They're one after another, and they're exactly the same thing, okay? So, for example, in yeast, which is a eukaryotic organism, the rDNA, which we call the ribosomal DNA, this is the DNA encoding for ribosomal RNA, we call it rDNA, is encodes for 150 copies of this repeat that's arranged in tandem on chromosome 12. So what you have is a 9.5 kb fragment that is just repeated one after another. Okay, and if you zoom into the fragment, what you see is that it's encoding for the rRNA genes here. Just disregard these other extra information. So what you need to know is that tandemly repeated genes are exactly the same sequence, and you have many, many copies. And a very good example of it is the rDNA. They usually are found in clusters in different regions of the genome. In humans, there are 400 copies of each rRNA gene that are scattered over 5 different spots in the genome, and I mean, basically, they are in different regions of the genome in multiple copies. So here's a question. Why do we need so many copies of the RNA? Why do we need so many copies? What does RNAs do? Ribosomal RNA. You learned about ribosome structure. They're components, they're integral components of the ribosome. If you don't have rRNA, you don't have ribosome. What do ribosomes do? They translate proteins. When do you need proteins? Always a good answer. When do you need proteins even more than always? In a developmental, think of a developmental timeline of an organism, an animal, from beginning as a single cell to becoming us. When do you think we're going to need a lot of ribosomes? Speak up. Embryo, because we're going to divide, right? You're going to make new cells. To make new cells, you need more proteins. To make more proteins, you need more ribosomes. So in a human cell, for example, during embryogenesis, we have rapid divisions. Every 24 hours, a cell divides and makes 2 daughter cells. So it's going to have to replicate itself completely in 24 hours. That's that amazing. Like, imagine me making another one of myself in 24 hours. It's a lot of energy, right? And it takes about 5 to 10 million ribosomes per cell to do that. And to make that many ribosomes, you need to make a lot of rRNA. And because the RNA polymerases have a certain speed, that they can only synthesize so many number of nucleotides per second, right? If you have only one copy of the rRNA gene, you're never going to make enough in 24 hours. But if you have hundreds of copy of that gene and you constantly transcribe them, then you can actually meet the demand, right? And so that's why you have. Go ahead.
So when you're not in a state of rapid division, do you just have like a bunch of excess of rRNA, or is it regulated?
Excellent question. Perfect question. That's exactly what a scientific question would be like. Actually, when you're not dividing, what happens is these genes get silenced. Okay, so what happens is their chromatin, which we're going to talk about in the next few lectures, the chromatin makes these genes compact, and squishes them in the nucleus, the RNA polymerases won't transcribe them, so they're going to remain silent. And in some cases, they might even recombine out, and you reduce the copy number. That usually happens in yeast, for example. Nice. Okay, so what else is there? So far, we only covered 25% of our genome, approximately. What's in the rest of our genome is, it used to be called a junk. We actually don't know if it is simply a junk, but it's repetitious DNA. So these are repeats that are coming from transposons. These DNA molecules that can copy themselves and propagate themselves in our own genome. So it's our own genome parasites, if you think about it. All right. So what kind of repetitious DNAs are in our genome? There's all kinds of different repetitious DNA, and they are in the non-coding regions of the genome. Non-coding means the regions that are not covered by exons, right? And in human cells and in larger eukaryotic cells, larger eukaryotic species with larger eukaryotic genomes, you have actually more non-coding RNA. It's not that you have a lot more genes, but you actually have much more non-coding RNA. And what we think is that because you can now regulate how those genes are produced. So, for example, here's the 80 kilobase region in humans covering the beta-globin gene cluster. And here's a corresponding region in corresponding 80 kb region in Saccharomyces cerevisiae, that's yeast. So each of these green boxes is a gene in yeast, and this in humans, the same region which contains lots of genes in the yeast contains very few genes because it's interrupted with lots of non-coding DNA. And some of this non-coding DNA, actually, most of this non-coding DNA is repeats. All right, here's another question. Let's do some thought experiments, or let's think, right? Why do we tolerate so much non-coding DNA? The smaller organisms like yeast and bacteria, they pack up their genes. In fact, in bacteria, some genes are overlapping. So one gene is transcribed in one direction, the other one gene is transcribed in another direction. So the genome is completely packed. It encodes for a lot of information. But humans, we have lots and lots of repetitious DNA, we don't even care. We can totally tolerate it. So why do you guys think that throughout the evolution, the animals like us tolerate a lot of repetitious DNA, and maybe smaller bacteria, they don't actually tolerate that? Any ideas?
Is it because, like, in DNA replication, we kind of lose some, so you get, like, a region that's, like, expendable?
But the expandable region contains no information. So you would be expanding repetitious DNA that doesn't really add anything to you, right? It doesn't encode for any genes or anything. Yeah. Go ahead. Mm-hmm. Yeah, go ahead.
It's because we're multicellular, so it can each region can specialize only in one function, while bacteria is unicellular, so it has to do everything all from one genome.
That's true, but that is really not explaining why we tolerate it, because we can have genomes just like bacteria, but we could use smaller pieces in different tissues as well, right? So that doesn't quite say it. Go ahead. Can you speak up a little? Well, that is a good idea. That's not quite what I was going for, because what I don't know is if the mutation rate actually is different. It might not be different because, that's right, it will be different because it's your chances of hitting an exon is going to be smaller in the larger genomes, right? But it doesn't quite explain the difference why bacteria then doesn't have lots of a large genome, right? They could have used the same strategy, but they're not doing it. All the way in the back?
Is it because of regulatory sequences?
That's right. A lot of non-coding DNA encodes for regulatory sequences, but repetitious DNA actually don't encode much of a regulatory sequence at all.
Is it because prokaryotes and simpler eukaryotes just been around longer from like an evolutionary standpoint?
Actually, prokaryotes have been around much, much, much longer than us.
Right. So is that
And how's that explaining the genome repetition difference?
don't prokaryotes have more compact, efficient genomes than we do?
Efficient is kind of along the way. So the idea is this. In evolution, what you have is you have random mutations, right? And then in a population, then one or few mutations actually give you advantage, right? And then that is going to now expand. And it's going to have lots of progeny, and it's going to take over, right? But there is a natural selection. There is a selection applied to the population. Now, for bacterial populations, the natural selection selects at the level of reproduction. That means if a cell has, for example, a higher division rate, that is going to take over the population because that's how they reproduce, right? You divide. And to divide fast, what do you need to do? You need to replicate your genome fast, right? Because if you want to make 2 copies of yourself, and if you have a huge genome made of all kinds of repeats, you're going to take a lot longer to replicate that. So the bacteria that have smaller genomes are going to have advantage over their cousins that have larger genomes because they can divide faster, right? But do selection in humans work at the level of reproduction? What do you think the selection would have worked on back in the day? Survival. You're running away from lions, right? You're not. It doesn't necessarily. It makes nothing if you can make lots of babies. They're not. They're going to be eaten by lions. Sorry about my horrible language, but it matters that you actually run faster, right? So there are different selection pressures on different kinds of populations and genomes. All right. Okay. So what kind of repeat DNA are we talking about here? So there is one class of repeat DNA called simple sequence DNA. These are usually short tandem repeats. They're 1 to 500 base pair. If they are about 1 to 13 base pair and they are less than about 150 repeats, these are called microsatellites. If they're a little bit longer, they typically have fewer repeat numbers. These are called minisatellites. The number of repeats of microsatellites and minisatellites in different humans is very different. So these sequences are used for DNA fingerprinting. They are usually not in genes. Most of these type of repeats are found in centromeres and near the telomere, and they have a role in establishing heterochromatin, which we're going to talk about in the next lecture. So there are different numbers of these microsatellites and minisatellites in different humans, and why and how, right? It is thought that this is due to the mistakes that DNA polymerase makes. When DNA polymerase is replicating the genome, it needs to make exactly the same copy, right? But DNA polymerases actually are not perfect. Sometimes they make mistakes. And one of the mistakes that they actually make during normal replication, for example, here is a microsatellite where you have GTC. This is a 3 nucleotide repeat, one after another, right? And so when during normal replication, imagine that this top strand is the new strand that's being synthesized. You should make exactly the same copy that's matching the parental strand, which is the bottom strand, right? But sometimes what our DNA polymerase do is it slips on the template. So it's going along making exactly the exact copy of the parental strand, but sometimes it slips backwards, okay? So when that happens, if this is in a non-repeat region, it realizes that it slipped backwards, because when it slips, the nucleotides that are being synthesized are not going to be complementary to the nucleotides in the back, right? So what you're going to have is a non-complementary region. Polymerase usually recognizes that and corrects it with certain correction mechanisms. But in repeats, when it slips, sometimes it doesn't realize that it slipped. And the reason for that is when it slips backward and the DNA that is in the new strand loops out, okay, and then the polymerase continues to synthesize DNA, and it makes a copy of the new strand here, right? So now what happens is that now you created a chromosome, you created new DNA that has one extra copy. So you have n numbers of copies in this person, and then now you have one more copy. Now, when this DNA goes through a second set of replication, what will happen is each of these strands are going to be replicated, and what you're going to have is you're going to have two daughter DNAs. One is going to have one extra repeat, and the other one is going to have the original number of repeats to begin with. So this is one way the number of repeats in the genome can expand. Why does this matter? Most of these repeats are in intergenic sequences, so they do not affect proteins, okay? But some of these repeats are inside proteins. And when you have a repeat of size 3 encoding for a certain amino acid, right? If you have repeats of 3 nucleotides, and if you have one extra, you're going to put one extra amino acid in that protein, right? So you're going to now affect protein structure. And does this happen? Yes, it happens. For example, here's a gene called RUNX2. It's a transcription factor, and there are 3 nucleotide repeats inside this gene and makes a stretch of glutamines. And depending on how many repeats you have, you're going to put that many number of glutamines in the protein. And this length of this stretch of glutamines alters the transcription factor activity, and what that does is it leads to facial phenotype differences because this gene is important for craniofacial features in humans and in dogs. And for example, here's a very nice study. What they did here is they took a dog skulls from certain times. So this is from 1850. 1921 and 1967, and what they did is they compared the facial features, and what they found is the breeders like a certain facial feature, right, and they keep breeding it to make that feature more prominent, and what they were selecting basically was they were selecting for the number of repeats in this transcription factor. So the dogs that have certain features had higher copy numbers or lower copy numbers of that repeat. So the repeats basically provides a lot of phenotypic differences within a species if it happens in a gene. And there are also repeat-based diseases where if you have certain number of repeats, the protein becomes non-functional, and that causes certain human diseases as well. All right. Because these repeats are in different numbers in each of us, we can use them for DNA fingerprinting. What does DNA fingerprinting mean? So in this case, what the technology does is it takes certain repeats in the genome, and it PCR amplifies these repeats with specific primers, right? Let's say there are 4 repeat regions, and each of those 4 repeat regions have different numbers of repeats. If you PCR amplify these repeats and run it on a gel, for each given human, you're going to get different length products, right? Because you amplify that repeat. So basically, let's say that in person A, you have a repeat region where you have 4 repeats, and you have PCR primers that are amplifying this repeat. Let's say you're getting a 50 base pair product. When you run it on a gel, you're going to see a 50 base pair product. In person B, you have, let's say, 8 of these repeats when you PCR amplify with exactly the same primers. Because this region has a higher number of repeats, you're going to get, let's say, 100 base pair product, right? So when you run all of these PCR products in a gel, you're going to get fragments of different sizes, correct? So let's say you're in a daytime talk show. Oh, no way. Okay, let's see here. What happened? Okay, let's say you're on a daytime talk show. They're trying to do a paternity test. You're the scientist actually doing it. Okay, they have three, you have a mother, and you have a child, and you have two father candidates, right? So what you do is you do DNA fingerprinting. You fingerprint the mother, you fingerprint the child, and you fingerprint the two fathers, candidates. And what you do is you compare the fingerprints of the child to the child to the father, right? And you look for similarities. For example, here, the child has this DNA length, this DNA fragment. So in her genome, there were that many repeats, and it turns out that the father number 1 also has that particular band, right? But one band is not enough. You have to do it at many different regions of the genome to have statistical significance. So what you do is you look for similarities. And looking at just the patterns, which one of these candidates might be the father? 1, right? But what you can also see here is that, for example, why does this father doesn't have this particular band here? Because it's coming from the mother, right? Remember, the child is taking her genome from the two parents, right? So the child is taking one allele from the mother, one allele from the father. So if a certain allele, that means like certain repeat number, if it's not taken from the father, you should see that in the mother, right? So if it's taking this allele from the father, you see it from the father, and you don't see it in the mother. The child has not taken up this allele because during meiosis, the mother is creating gametes with half the number of chromosomes, so the child never inherited that certain allele, right? So the pattern is not going to match exactly. It's going to be a mixture of the mother and the father. You're not going to see all of the alleles, but the child should be a combination of the alleles that you should see in the mother and father, right? And so that's how you would do that. And this kind of DNA fingerprinting is used in criminology a lot. For example, any type of criminal identification that is based on DNA typically uses DNA fingerprints. For example, if there is a crime and you have a victim and you have the suspects, and you take some samples from the victim and you can compare it to the suspects, and you can see which of these suspects' DNA fingerprint matches to the victim. And because all of our genomes are very different in terms of repeat numbers, the chances of a certain pattern to match exactly is very low, except one case that it could be that this test could totally fail. What could it be? Identical twins. So if you have an evil identical twin, then you're in total trouble, right? Okay, so basically the simple repeats are used for DNA fingerprinting. Any questions about this? No. Okay, so these are short repeats, the simple repeats. There are longer repeats. These are called interspersed repeats. They are 100 to 1,000 base pair long, sometimes even longer. They're found in prokaryotes and eukaryotes everywhere. They're called the mobile elements because they jump from one site in the genome to another. They're lost very slowly from the genome because they're very good at replicating themselves. They're very good at jumping when there is trouble. Okay, so they were first discovered by Barbara McClintock. She discovered the mobile elements in 1940s, and this was really controversial at the time because the genome we know is that contains the information. So it was very difficult for the scientists to imagine that the genome would change so rapidly by little elements jumping from one side to another. So people resisted this idea a lot. So even scientists can be kind of conservative sometimes. And then, of course, quite a few years later, people replicated her results and realized that there are mobile elements everywhere, and she was properly recognized. All right, so there are two different types of mobile elements. We're going to talk about them. The two classes are, we generally classified them based on the mechanism by which they jump from one place to another. So we have DNA transposons and we have retrotransposons. The DNA transposons, basically what you have is a DNA fragment that is the transposon. It gets excised out from the donor DNA, so that is the original location, and it gets inserted into a new DNA site, that is the target site, and what it does is it gets in a different site. So jumps from one site, inserts itself to another site. Retrotransposons are not named based on their fashion sense. They're called retro because they get transcribed back into mRNA, back into RNA before they jump. Okay? So what happens in this case is that you have the transposon at the original donor site in the genome. This transposon gets transcribed by RNA polymerase and becomes an RNA. This is an RNA intermediate. And what happens is that there is an enzyme called reverse transcriptase. So it does the reverse of transcription. It makes DNA out of RNA and makes that DNA which gets inserted into the new target site. Okay? So how you can remember these two mechanisms simply by thinking that DNA transposons mechanism is a cut and paste mechanism, right? And the retrotransposons mechanism is a copy and paste. It copies itself because you have exactly the same DNA in the donor site. It doesn't get lost, right? You make a copy of it in a new target site. All right. Here's a DNA transposon in bacteria. They look something like this. They're made up of a few KB fragments. In the middle of the fragment is the region that encodes for proteins. So this encodes for a protein called transposase that transposes the DNA, inserts the DNA into a new site. It is surrounded by inverted repeats, typically containing promoters or other regulatory sequences. And each transposon is surrounded by a short target site direct repeat. So direct repeat is basically a 5 to 11 base pair sequence that is exactly the same at the end of the transposon and at the beginning of the transposon. And this is basically a signature of the transposition reaction because of how it happens. And this is how it happens for the DNA transposon. What you have here is that you have your transposon at the donor location, so it's in the original location, and you have a target DNA. Okay, the first thing that happens is the transposase, that enzyme that's encoded from the transposon, makes a cut at the target site that is staggered. Staggered means two cuts that are offset from each other in the two strands. Okay, so that's making a staggered cut at the target site, and it also makes a blunt cut at the donor site. Blunt cut means you're cutting the both strands exactly the same location, right? So you have a blunt end. What happens next is it takes that DNA fragment that's blunt cut and it inserts it and ligates the two ends to the target site. Okay, but because the target site is cut staggered, what will happen is you're going to have short stretches of DNA here and here that's going to left single-stranded, correct? And those single-stranded DNA is going to be filled by the DNA polymerase in the cell and get ligated. And when that happens, you basically have a direct repeat at each flanking end of the transposon, and that's the signature of the transposition. So the mechanism of those direct repeats is due to the staggered cut that is made at the target site. Does that make sense? Any questions? Okay. Since this is a cut and paste mechanism, that is, you're basically moving the original donor DNA into a new location. How do the DNA transposon number increase in copy? Okay, so here is an interesting question to think about, and the answer to this question is in your textbook. So what I'd like you to do is, when you go home, look at your textbook and read about how this might happen. Okay. All right. So retrotransposons reverse transcribe. So they go through an RNA intermediate. They're typically longer than DNA transposons. Again, the way that they're organized is kind of similar. You have a middle region that's protein encoding. So it encodes for proteins such as transposase and other proteins that help this transposon move. And you have long terminal repeats that usually contain promoters for the genes in this protein coding region. And you have target site direct repeats. So long terminal repeat transposon make about 8% of the human genome. They're retrovirus-like elements. Instead, retroviruses encode for envelope proteins, the capsid proteins, so they can burst out of our cells and infect everybody else, right? But these retrotransposons are happy in our genome. They don't burst out because they do not make these capsid proteins. What they do is they live in our genome by jumping from one side to another. The long interspersed elements in humans make up most of our genome. So the long interspersed elements, they're called lines. There are 900,000 copies or so, which makes 21% of our genome, right? So this is amazing. The full-length lines are about 6 kb, and you also have short interspersed elements. These are kind of like lines, but they're shorter versions of it, and they make up about 13% of our genome. And because they can jump from one side to another, they can cause mutations in our genome. So if it jumps, for example, by accident into an exon of a protein coding gene, it could disrupt that protein and cause problems, right? So a lot of our mutations also that cause disease are from lines and signs. How do they jump? Their jumping is a little bit more complicated and more interesting. So what happens is the first step is the creation of an RNA from the transposon. Okay, so this is called the line RNA. So we're talking about line transposition here. The RNA is transcribed by RNA polymerase II. So a polyadenylation occurs. So you have an A tail at the end of the mRNA. So what the ORF2 protein, this is a protein that is transcribed by the transposon, does is it takes that mRNA and goes to a target site. And it lines up that mRNA with the target site and makes a scattered cut again. So it makes these little nicks, right, offset from each other. Now, the end of this nick here, the end of that DNA fragment, is the 3' end of the DNA fragment. So that end is taken up by the reverse transcriptase. This is the enzyme that copies RNA into DNA. And what it does is it uses the information in this RNA, it makes a DNA copy, okay? As it copies that RNA, the DNA gets extended, and the cut site, the staggered cut sites start opening, and it makes a copy of this DNA, and when it reaches the end, the DNA polymerase fills up these gaps, so these gaps of single-stranded DNA of the target site, and again reaching in these gaps and making a direct copy. Now, what we have here is a copy of a DNA that is hybridized to RNA, right? So what happens is that the cellular enzymes that think that this is not right, which is not right, you usually have a double-stranded DNA, and what they do is they take the 3' end of this DNA and basically remove the RNA away and make a copy of this DNA now and reach to the end and get ligated. And what happens at the end is you have an exact copy of the line DNA that is inserted into a new site, right? So that's how it goes through a intermediary to copy itself. Does that make sense?
So the repeating sites, like that determine where the transposon is, would those include elements like enhancers that might, like, before that gene but further up in the genome?
It could. Depends on, so the LTRs, the terminal repeats, they are transcribed, so they are going to end up in here. Basically, this is where they're going to end up, but not always. And sometimes they do include enhancers, but not often. They usually include just promoter sequences to make sure that they can get transcribes themselves. Does that answer your question?
Yeah, but if it didn't include the enhancer, wouldn't that have like regulation issues?
So enhancers are usually for genes that are going to be expressed in different tissues. These DNA transposons and retrotransposons typically don't have enhancers. They do not need enhancers. In fact, the gene's transcription does not need enhancer. What it needs is a promoter that will be strong enough to recruit RNA polymerase II. What enhancers do are these are DNA elements that regulate the expression of a gene in different tissues. So they turn off the promoter or they turn on the promoter in certain tissues. So these type of retrotransposons, they do not typically contain enhancers.
Sorry, could you explain again the purpose of the transposons and the retrotransposons?
There's no purpose. What's our purpose in life? Their purpose is propagate themselves. They're selfish, just like us.
So are they, but they're not a virus.
They're not a virus because they lost their capsid proteins. They're not independent. Think of it as a parasite. It's a parasite of our genome. They live inside us, in our genomes, and they propagate themselves. Does that make sense? It's a nice sci-fi kind of thing, right? Any other questions? All right. So, the signs, these are short interspersed elements, are just like lines, but they're truncated. And they're usually truncated at their 5', okay? So, what that means is the beginning of the mRNA is missing. Looking at this mechanism that I pasted here, can anyone guess why the signs are truncated at their 5'? What could be the basis of that truncation? All right, let's look here. The first step, one of the first steps of the transposition is taking up that line RNA and making a copy of that RNA into DNA by this enzyme called reverse transcriptase. And I'm telling you guys that the 5' ends of the lines are usually truncated, forming signs. What could be the basis of that? What property of this reverse transcriptase could have led to that? What if this reverse transcriptase is not a very good enzyme? and it actually stops before it finishes, then that would create a 5' truncated line element, right? Because this is the 5' of the element. So you'll never copy this side of the RNA, you'll only copy that side, right? So you make a shorter one, and that shorter one gets integrated. And that happens because reverse transcriptase is a crappy enzyme. That means it doesn't do its job always. It can't go all the way to the end. It falls off. That means that terminology in the polymerases is called processivity. That means how long can you process before you fall off. And reverse transcriptase, compared to DNA polymerases that replicate our genome, is a crappy enzyme. It doesn't do a very good job. It doesn't have high processivity. All right. Again, this thing. All right. So because of this, we have lots of short interspersed elements called signs in our genome, which make about 13%. In humans, 70% are called ALU sequences. This is simply because these short sequences contain a restriction site for ALU1, and that's how they were first identified. And these short ALU sequences are typically transcribed by RNA polymerase III, and they're also partial sequences. All right. Transposons can have a big effect on how our genomes evolved. In fact, they create so much diversity in our genomes due to their ability to move around, right? So we not only have diversity due to mutations, we can have diversity due to transposon activity in our genome. How does that happen? So here's an example from your textbook. Imagine that you have two DNA transposons, okay, that are surrounding an exon. Now, the transposase usually makes a blunt cut on each end of each DNA transposon, right? But nothing is perfect in life. And sometimes these proteins also make mistakes, okay? Let's imagine now this protein, instead of cutting in each end of this DNA transposon, it cut from this end of this DNA transposon and cut on the other end of the neighboring transposon, okay? Because these sequences are exactly the same, it can make a mistake and mix up. And what that will create is it will create a long DNA fragment not only containing the transposon sequences, it will also contain that exon in the middle of them, right? And now when the transposase enzyme inserts it into a new target site, you not only made an insertion of your transposons, you also inserted now the exon in there, okay? So what happened is now you put a new exon inside an existing gene, for example, and when this gene gets transcribed and spliced, you might make a protein containing this exon, this new exon, and that exon, and that exon, right? So now you generate a completely new protein with this exon by this transposons bringing a new exon. All right. This kind of mistakes also happen for retrotransposons. For example, sometimes when the retrotransposon is being transcribed, the RNA starts getting made from the beginning of the line element. It goes to the end, but sometimes RNA polymerase cannot recognize a weak termination sequence, so it doesn't stop. What it does is it continues transcribing and makes a longer RNA transcript, right? The enzymes that are taking this RNA transcript does not recognize the sequence. What they recognize is the transposon sequence containing RNA with a poly A tail, and what they do is they take up this RNA and insert it into a new site, And because this RNA contains this last exon, right, so this is the 3' exon, now when it gets inserted into a new site, it actually brought in that exon with it, right? Does that make sense? So the DNA transposition and mistakes in it can cause exons shuffling from one site to another. This is also called exon shuffling. All right. The transposons can also make changes, retrotransposons make changes in the genome by accidentally picking up a polyalylated RNA that encodes for a protein itself. Okay? So what it could do is sometimes it can take that RNA, insert it in a new location. When you have two copies of exactly the same genes, and there are mutations randomly happening to these two genes, sometimes you do not need both of those copies, okay? Sometimes the second copy of the gene accumulates mutations, and if these mutations allow for newer functions, different functions, it would adapt to those different functions. But sometimes it doesn't need to do that. And what happens is that it accumulates lots and lots of mutations and becomes non-functional. So it doesn't encode for a functional product in any case, but the sequence looks very similar to the original gene, right? And these are called pseudogenes. They're not quite the genes themselves. They have lots of mistakes, but they still look like genes, right? And so these are called pseudogenes. For example, let's say that this gene, called the translated region gene, was accidentally inserted in the genome and went through lots of mutations. And so when you, for example, sequence the genome of a species and you saw a gene that looks like another gene but has lots of mutations and it has short direct repeats that are flanking the pseudogene. What would your guess would be? If you're the researcher that is trying to guess the origin of the pseudogene, what would your guess be? What kind of mechanism might have led to this pseudogene? I just told you, like 2 minutes ago. Think about it. It has short repeats, so it's a transposon, right? You say, yeah, I know, that's a signature of transposon insertion. So it must have been a transposon thing. And you say, what are the different types of transposon? I know there's a cut and paste, there's a copy and paste, and which one do you think is giving rise to the pseudogene? Cut and paste or copy and paste? Copy. What's the evidence for that? DNA transposons can do exactly the same thing. That's the question I asked for everybody go look into their textbooks, because they can actually give rise to a second copy through a different mechanism. Okay. Another guess. It's screaming there. Once I tell you, you guys are going to say, oh my God, I didn't see that. It's screaming, saying, I'm the evidence. It's because these introns are no longer there, right? So when the copy and paste happens, it goes through a mature RNA intermediate. That means the gene gets transcribed, the introns get removed, and it becomes an mRNA, and the transposition machinery accidentally picks up that mRNA and inserts it to a new site. So while the sequences of the exons you will recognize in a new site, you will not recognize any of the sequence of the introns, right? So that's how you know it went through an intermediate. Any questions about that? All right. So the repeats make up a huge portion of our genome, and there is another class of DNA, which makes about 25% of the genome, that we call them unclassified spacer DNA. So a lot of the research now after human genome sequencing is trying to figure out what is in this unclassified spacer DNA. We know that there's a lot of regulatory information. That is, the enhancers are there. There are lots of enhancers in the intergenic regions that control gene expression in different tissues. But also what we have recognized in the past decade or so is that some of these unclassified DNA encodes for RNA products. These are functional RNA products that we didn't know existed before. Okay? So we know some of them. We know that here's a functional RNA product. That's RNA, tRNA, small nuclear RNAs, and we now know there are microRNAs. These are RNAs that gets processed into small pieces, and they are used to silence genes, which you're going to learn later in the course. There are long non-coding RNAs, such as exist, which is important for exon activation, which we're going to talk about, and a long list of RNAs that we know we started to understand their function. Okay, so some of that unclassified genome is actually starting to be classified as well. All right, in the human cells, we have the nuclear genome. That is our genome, right, which we always recognize as the human genome. When we think of the human genome, we think of the DNA inside our nucleus, right? But there's also non-nuclear genome. Each of us have a mitochondrial genome, right? And that is the DNA that's in our mitochondria. So as you know, some organisms, all organisms have, all eukaryotes have mitochondria, prokaryotes too, I guess. And some organisms also have chloroplast, and these organelles are thought to have evolved by taken up by an ancestral cell, and they basically evolved with our cells with time, and they become specialized organelles for special functions. And they still contain some of their genomes that encodes for genes that are some of genomes containing genes encoding proteins that remain in the mitochondria and remain in the chloroplast. And there are also some genes that encode for proteins that cross that barrier. But so far, what we know, if you look, for example, at the proteins that are inside the mitochondria and look where their genes are located in the mitochondrial genome or the nuclear genome, what you find is that for each complex of proteins, some of these genes are located in the mitochondria and some of these genes are located in the nuclear genome. So the two genomes are collaborating to make these mitochondria structures. All right, so I have sometimes I will have links for useful animations at the end of the lecture, so you guys should go ahead and watch these. And also read the textbook. And just like Professor Siegel, I will have study questions at the end of the lectures, and you can find these in the PDFs. So go ahead and study them and discuss with your recitation structures, etc. One more note about office hours. I do not have a set office hour during the week, but if you simply email me, I will fit you into my schedule in a few days. So I'm very accessible. Don't worry about it. Just shoot me an email.